This is online lecture 13 for Bio 240. This week we're talking about chromosomal rearrangements. As you know, within a species there is a defined number of chromosomes which contain the same genes from one individual to another. For example, in humans there are 23 pairs of chromosomes. And if we stain the DNA and look at the light-dark bands, the banding pattern is generally the same between one person and another. However, we know that we're not genetically identical, except for monozygotic twins, and this is because of mutations found in all individuals. So far we've talked about mutations in genes at the level of different base pairs, which lead to different alleles. But today we're going to be talking about mutations in chromosomes. These are chromosomal rearrangements. These are changes to the structure of chromosomes. So unlike a single nucleotide change, chromosomal rearrangements involve large-scale alterations to the chromosome. And there are 4 types that you should be aware of. The first are duplications. This is where a large segment of the chromosome is duplicated within that same chromosome. The second are deletions. And this is where a large segment of a chromosome is deleted. It's just missing from a chromosome. The third are inversions. This is where one segment of a chromosome actually gets reinserted in an inverted manner. And the fourth are translocations. Translocations are where one part of a chromosome gets inserted into a different chromosome, not within the same chromosome. So these first two we're not going to talk about in this lecture. You should refer to your textbook. Make sure that you understand them. Today we're going to focus on inversions. And then on Thursday we'll talk about translocations. So we'll start out talking about what an inversion is. And I've drawn here, if this is a chromosome, we can say that somewhere along the chromosome will be the centromere. And then the chromosome, we could just imagine that it's broken up into segments along its length. And we can arbitrarily just call those segments A, B, C, D, E, F, G, H, I, and J. So we've just broken this chromosome up into different segments, and what happens with an inversion is that sometimes there will be a break in the DNA sequence along a chromosome. So if there were a break here and a break here, a lot of the times when the DNA breaks, we have DNA repair mechanisms, and it will just repair itself in the same order that it was originally. But sometimes you'll end up with a chromosome where that segment that broke out is actually inverted. And so if we think about that inversion, you can see here that this region of the chromosome where the breaks occurred has been inverted. And so we call this an inversion. And now there's 2 types of inversions. So inversions can be what we call paracentric. And the inversion I just showed you here would fall in that category. So paracentric means that it does not involve the centromere. versus pericentric. And that's where the inversion itself happens, including the centromere. So again, the example I showed you up here with this inversion, that's paracentric, meaning next to the centromere, but it doesn't include. Peri is around, so peri would be an inversion that somehow included the centromere. So you may be thinking, what's the big deal? We have a piece of DNA, it's inverted, but it's still, the whole chromosome is there, right? So what are the consequences of an inversion? And again, here is an example of our inversion. Well, the first thing is that it can, depending on where that break is, the break in DNA may be able to disrupt a gene. So imagine that right here in this sequence of DNA is coding for some gene. And when that sequence breaks, you get a break in the middle of that gene. And so that gene, even when it gets reinserted and you repair that break, if that sequence is now at a different place, you're not going to be able to match up the correct sequence with to make the full gene, and that gene will no longer be functional. So that's the first consequence. And that doesn't always happen because there are large areas of the DNA sequence that are non-coding, but if it happened to be in the middle of a gene, either one of these breaks, that could disrupt gene function. The second consequence is called positional effect. So even if there's not a gene directly in the region where there's a chromosomal break, there are genes within the inverted section that now, in this altered structure, the genes that were down here used to be over here in the chromosome. So they have a different location relative to other genes and relative to other promoters and regulatory regions within that DNA. So they might have altered regulation. So we have altered regulation of genes in the inverted sequence. Or of genes outside of the inverted sequence, but whose regulatory or promoter region happens to be within that inversion. So again, these are positional effects because it doesn't affect the function of the gene itself, it affects the expression or the regulation of that gene. So this can have an effect like when a gene is expressed during development, or when during the cell cycle it might be expressed, or which cell types might express a certain gene. So these are all because of altered regulation. And then the third effect that can happen from these inversions is when a cell that has these inversions gets ready to go through meiosis to form gametes, you can end up with fewer viable gametes. And so, in humans that would translate into reduced fertility. And this is true for individuals who are heterozygous for the inversion. So most of the time when an inversion happens, it only happens in one chromosome. It could rarely be the case that an inversion is passed on, and you'll end up with two copies of that chromosome, two homologs that both have the inversion, in which case they match each other, so you would only be dealing with these first two effects. However, most people, again, are heterozygous for an inversion, or most individuals are. So how would this affect the viability of the gametes? So the problem arises during meiosis, specifically during prophase 1, when the two homologs try to pair. So remember that during prophase of meiosis 1, you have homologous chromosomes lining up. That is, remember, every cell has two copies of chromosome 5, for example. So those two copies of chromosome 5, the one from the maternal side and one from the paternal side, are going to line up, and areas where there's a lot of homology between the sequences will pair up. So I'm just going to draw that out here. So here would be our two homologous chromosomes. One of them has the inversion. So remember that before entering meiosis, the cell undergoes DNA replication. So I've drawn here the two sister chromatids for each of these chromosomes. And then during prophase 1, they're going to pair up where they have similar sequences. So the centromeres can pair up, and all along these regions, there would be no problem. But when we get here, H and F, these two segments are not going to be homologous, and they won't be able to pair up. And then same thing over here. So what does the cell do? How does the cell cope with this type of problem? So I'm just going to go back to shorthand notation here, so instead of drawing them out as big fat chromosomes, I'm going to draw them out as lines. And so this is the same thing represented again. I just have our two sister chromatids connected by the centromere. And here's the other homolog with the two sister chromatids connected by the centromere. And in this region and this region, that's where the inverted segments cannot pair during prophase 1. So what's going to happen is the chromosomes will organize themselves in a way that allows pairing to occur. And they do this by forming a loop, which is called an inversion loop. So if we take just the normal chromosome here, what it's going to do is that region that can't pair up, it's going to form a little loop. And if we draw in the sister chromatid with it. So there's our two chromatids. So we have A, B, C, D, E, and now F, G, and H have looped out, and then we have I and J down here. And now what's going to happen with the second chromosome, I'll just draw it in a different color here, it's going to actually twist itself so that it can line up homologous segments. So if we have A, B, C, D, E, and now see how where the twisted region is, we have H, G, and F, I, and J. So the inverted segments, now because it's twisted, line up with the correct segments from the first chromosomes. Now our homologous chromosomes can pair. And if the pairing goes like this and there are no crossing over events within that inversion loop, then meiosis can proceed as normal, and you're going to end up with your 4 gametes. If you remember, each of these chromatids will separate into a gamete, and what you'll end up with are two gametes that have the normal chromosomes. They came from the top homologue. And then you'll have two gametes that still have that same inversion. But as long as none of those genes that were disrupted from the inversion are important for gametogenesis, these gametes will be viable. So again, that's if there are no crossing over events. But let's look at what happens if there is a crossing over event within that inversion loop. So here are our two homologous chromosomes lined up. And within the inversion loop, let's say there's a crossing over event that happens here between G and H. And so remember that the crossing over happens between the two chromatids that are closest to each other. So the outer chromatids are going to stay the same. So we're going to end up with the outer chromatid from the top chromosome is going to be normal. Again, I'm writing out what the gametes will look like. Then we can look at the bottom chromatid where that didn't get the crossing over. And like we saw before, this one will just have the inversion. But it will be fine. But take a moment to trace through the chromatids in the center. And let's look at the bottom of the black chromatids first. So we're going to have A, B, C, D, E, and as we go around here, we have F, G, and then we're going to switch over, and we'll get a crossing over, and now we're going to start with the green one, and we're going to have H from the green one, and goes back to E, D, C, B, A. So if we draw that out, we have A, B, C segments, the centromere, D, E, F, G, and here's where the crossing over event occurred. And so from G, we went H, E, D, C, B, A, with another centromere there. So look at this chromatid, this chromosome that ended up in one of the gametes. Now we have two centromeres, which that alone will mess up the rest of meiosis. We also have a duplication of several segments, and we have deletion of other segments. So this gamete is going to end up being not viable. So now let's trace the last sister chromatid that was part of this homologous pair. And remember that this region already got tacked on to the wrong chromosome because of the crossing over in the loop pattern. So the last one we have, if we trace it out, would be J, I, and then it's coming up here through F, G, Then it's going to cross over, and it's going to have H, I, and J from the black. So again, that would be I, J, F, G, and then the rest of it would be H, I, J. So this one we would call acentric because it has no centromere. It also has major deletions, and it does have duplication of the I and J region. So again, that's too many problems. This chromosome will not make it through meiosis. It will be non-viable. So from this meiosis, where we did have a crossing over event that happened within the inversion loop, only 2 out of 4 gametes survive. And the 2 lethal gametes are the recombinants. So the offspring that you get from an individual who has an inversion are going to be more parental offspring and fewer recombinant offspring. So this example I showed you was with a paracentric inversion. Let's go on and look at what happens with a pericentric inversion. So when we're talking about a pericentric inversion, let's start out with our same chromosome that I started originally, and instead of the DNA breaks being between segments E and F and H and I, what if we had DNA breaks here and here, between A and C, B and C, and E and F? Now, if that segment were to be inverted, you're going to end up with so again, the centromere is a little bit offset, and you have a segment of your chromosome that is inverted. So there's the inversion. So this is why we call it pericentric, because see how it's around the area that has the centromere. So it involves the centromere. And the consequences for pericentric inversion are the same that we saw before. You may have disruptions in gene function if the breaks happen within a gene. You can have position effects with altered regulation of genes that are within that inversion region. Or you can have problems during meiosis. And for pericentric inversions, these problems during meiosis are a little bit different. So let's take a look at the details of what happens here. So I've drawn out for you how these 2 chromosomes, again, we're talking about an individual who's heterozygous, because in a homozygous individual, meiosis can occur normally. So this is in a heterozygote during prophase 1 of meiosis, where the chromosomes are lining up, and you'll see that the same thing, we get an inversion loop forming, and the inverted chromosome has to twist on itself in order to allow the centromeres and the homologous regions to line up. But by doing that now, we are able to line up homologous regions of the chromosome. So I encourage you to make sure that when you draw this out, you see how this looping action is occurring and giving rise to the homologous pairing that's happening. So as we saw with the paracentric inversion, as long as there's no crossing over event, meiosis can proceed as normal. You'll end up with 2 gametes. So again, this is no crossing over. Your gametes, you'll have 2 with normal chromosomes, and you'll have 2 with inversions. But all will be viable. But I just copied it down here. So now what if there is a crossing over event that occurs somewhere within the inversion loop? And we'll just put it right here between D and E. So now what's going to happen during meiosis? What gametes are you going to create? So pause the video here and try to draw out the chromosomes for each of the gametes that would be produced from this crossing over event within an inversion loop. And here are the 4 chromosomes that would be present in the gametes that result from meiosis after this crossing over event within the inversion loop. So we will end up with 1 normal, 1 inversion, and these would both be viable, as we've seen before. This third one here, notice it still has a centromere. However, we have duplication of the A and B region, and we have deletion of everything after the F, G, H, I, and J. So those are pretty significant, and this one won't be viable. For the fourth one, we have still 1 centromere, but now we have a very long duplication, and we have deletion of the A and B region. So both of these will be non-viable. As we saw with the paracentric now, we end up with 2 out of 4 gametes are viable, and the lethal gametes are, again, the recombinants. So to summarize, the take-home message is that for inversions, you get disruption in gene function at the area of the DNA break if it happens within a gene. You get positional effects that alter regulation of genes that are in and around the inversion. And you get reduced viability of recombinant offspring. So I'm going to leave you with a thought question, which is, how will an inversion affect a gene linkage analysis? And I want you to think about this for genes. Let's say you were looking at the linkage of 2 genes within an inversion, or 2 genes on either side of an inversion. And how would this inversion affect your gene linkage and your ability to map those genes onto a chromosome?